Hello and welcome to this video. Today we're going to be learning about aseptic technique in the laboratory, as well as how to perform a serial dilution. We use aseptic technique in many different settings to prevent the contamination of microorganisms. Within food science, this is used to avoid spreading pathogens. Now we're going to walk through the steps for transferring a bacterial culture to a medium without contamination. The inoculating loop is what we're going to be using today to transfer bacteria. The inoculating needle can also be used. An alcohol lamp will be used to heat and kill microorganisms. You want to start by first spraying your hands and lab bench with 70% alcohol to kill the microorganisms. If you use 95%, it may evaporate too quickly before killing the microorganism. We're now going to transfer bacteria from a liquid broth to another liquid broth. Begin by sterilizing the inoculating loop and pick up the culture and pass the lid through the flame, followed by the mouth of the flame. Dip the loop into the culture, then flame the mouth, replace the lid, and flame it again. Now you want to pick up the new broth, flame the lid, pass the mouth through the flame, and then transfer the culture with the loop. Flame the mouth, and then the lid, and then you're going to want to flame the loop until it's red hot. Now we will move the culture from a liquid broth to an agar plate. Start by flaming the loop until red hot, flame the lid, and then remove, flame the mouth, and insert the inoculating loop. Remove and flame the mouth, replace the lid, and flame the lid once more. You want to keep the plate close to the flame, and you're going to spread across with the inoculating loop, and then you're going to want to flame the loop afterward. Next is moving from a solid plate to a liquid broth. Sterilize the loop, and you want to touch the loop to a spot on the plate without a colony to cool it down and prevent from killing the colony. Then you're going to pick up a single colony while keeping the plate near the flame. Then you're going to heat the lid of the new tube, take the lid off, and flame the mouth. Then you're going to dip the loop into the broth, and then heat the mouth and the lid again. And then you're going to want to flame the loop until it's red hot. And finally, we're going to move a single colony from a solid plate to another plate. I'm going to start by sterilizing the loop at the flame, then touch the loop to the plate to cool it down, and then you're going to pick up a single colony. And be sure to keep the plates near the flame, and pick up the new plate and spread the colony onto it, and then afterward, you're going to want to flame the loop to sterilize. The final step of aseptic technique is to spray down your hands and lab bench with 70% alcohol once again to be sure that you kill any remaining microorganisms. Next, we'll be performing a serial dilution. We use this because most broth cultures will contain way too many microorganisms to count. If we dilute them serially, we can calculate how many colony-forming units are present within the broth culture. We will start with 7 test tubes labeled from 10 to the -1 to 10 to the -7. They will be filled with 4.5 milliliters of saline solution. We'll be using a micropipette set to 500 microliters to transfer our cultures to these tubes. Spray down the lab bench and your hands with 70% alcohol. To pick up with the micropipette, press down to the first half mark, then insert into your culture and release. To eject from the micropipette, press your thumb all the way down. To start, you want to pick up your micropipette and put on a sterile tip. Take your first broth culture, spin it on the vortex. You're going to want to flame the lid take the top and then flame the mouth as well. Then you want to insert your micropipette to pick up part of the culture. Then you're going to flame the mouth once more. Then you're going to pick up your new culture that you'll be diluting into. Go ahead and flame the lid and flame the mouth as well. Then insert your micropipette and eject the material. Flame the lid once more, and then you're going to want to eject the tip from your micropipette. You're going to want to keep diluting from the first to the second one. Go through the same steps that you used for the first one, then go from the second to the third, and so on until you finish 7 test tubes. Now you're going to want to spray down your lab bench and your hands once more with that 70% alcohol to be sure all the microorganisms are killed.